video, I'm going to just do an overview of the research that I'm currently working on. What I am working on is a computational project where I develop homology models about Plasmodium proteins as a way of screening potential drug targets. So the way this works is I get a sequence, I put it into a server that is able to generate a theoretical model similar to what you see on the screen here, and then I can pair it up with some ligands that are potential drugs that will then provide an output to find out which bind more favorably to this protein's active site. So the picture that you're seeing on here on the screen, this is a hypothetical Plasmodium protein called hypothetical because we don't know the specific function of it just yet. It does, however, have a protein tyrosine phosphatase domain. So what we believe it is that it functions as a phosphatase somewhere within the developmental process of a Plasmodium parasite. And these parasites, as you know, they are the causative agent for malaria, which is still a major problem around the world. Now, the way that I get to this protein here is that I first begin by going to one of the genetic databases that are freely available online. So there's Plasmodb. This is one that focuses specifically on Plasmodium proteins, so the Plasmodium genome rather. So all the different species of Plasmodium that cause malaria in a variety of different species, their genomes are all annotated here within the database. There are a bunch of other databases like GeneDB, ToxoDB, GiardiaDB that deal with other types of parasites, and you can always look up those. There is also another database that is known as GenBank. This is another large publicly available DNA database. This one provides DNA sequences, protein sequences of a variety of different species, not just looking at Plasmodium. And you can do the same thing where you can search a specific name. In here, you can look either for a nucleotide sequence or for protein sequences. There are a variety of different criteria you can use when searching. You can see here like protein clusters, protein nucleotide, protein family models, structure taxonomy. There are all of those. Now, the way that I start with this project, I can develop a strategy as it would show on here. I currently have a strategy that I've been working on in order to do a search. This search will break down a bunch of different criteria to help you narrow down the genes to a specific set of characteristics that you would like to investigate. But what I did first is I first did a search where I looked for phosphatases, narrowed it down to the different Plasmodium species, and you can see that there is one down here, PBX 091020. That's the current one that you saw pictured in the video there. Once I went over here and got this sequence, I can now look at all the features. What do we know currently? It will provide references as well. This provides a location within the genome of where you find it. You can find a structural analysis as well, where there is some structures showing you what type of theoretical models of the protein that are available. You can also go down, find some more references. Let's see, we have protein features and properties. A lot of this, let's see if we can pull up some information there. Tell you, there we go. So you can go over here and see it has a protein tyrosine phosphatase domain. That's from the superfamily database, InterPro. Again, verifies that. And then you can see in the concern domain database, a DSP. That is abbreviation for dual specificity phosphatase. Now, what I can do when I go to a database like this is I can go to sequences, and you'll notice that you have the predicted protein sequence as well as the RNA and the genomic sequence with the introns that are highlighted. So as you remember, within gene expression, introns are removed during the processing. The exons are spliced together. That gives you this mRNA sequence, and then that is translated into a protein. And so all of that information is provided over here. and we're able to copy this. So I could do something like copy into the clipboard. And if I want to make a structure in order to analyze it in further detail, the tool that I've always liked to use is the Swiss model. It's given me very good structures. I just paste my target right into there. So already it recognized it in the format. And if I would like to, I can add a hetero target if I would like to see how it might bind to something, but I can just go ahead and build a model. I might just call this one, I'll just name it the protein. So I'll just put PVX underscore 090120. And I'll leave the email blank for now because we'll build it up within here. And I can just go and click build a model. It usually doesn't take too long. What it is doing at this point is it is scanning the repository of all different proteins that have had structures solved, and the algorithm is able to predict potential folds and also domains and give me a theoretical confirmation of this protein based off of what we know about how other proteins are formed. It's a very intelligent AI type of program. It's been around for a very long time. And what we know about proteins, we know that certain amino acids, when they're beside other amino acids, will move and form bonds in a specific pattern. When we look at that over many different proteins, we will be able to get algorithms that will then predict what other proteins that we don't have the structures will look like. And so that's what this is doing right over here. It's running the project, and since I ran that project already, the output looks like this. This is the one that I gotten a bit earlier when I ran that. I just wanted to run the process just to let you see. But that's an overview of the homology modeling process. The next few videos, I'm going to go into some more details about how we do further structural validation as well as sequence analysis to verify specific features that we see in our homology models.